Hello everyone. In this class, we'll learn about cell organelles. Mitochondria. Mitochondria are the powerhouse of the cell. They are cigar-shaped organelles whose shape, size, and number vary in different tissues of the body. They are most abundant in cells that have high rate of metabolism, as we see in case of liver, cardiac muscle, etc. Structure of mitochondria consists of two important components, that is membranes and cristae. Mitochondrial membranes. Electron microscopy shows that each mitochondria has two layers of membranes, the outer and inner membranes. Outer mitochondrial membrane. This forms the continuous envelope of the organelle. Outer membrane consists of mostly phospholipids and cholesterol and contains a specific membrane protein that forms porine. Porine are the channels that permit substances with a molecular weight of less than 10,000 to diffuse freely across the outer membrane. Inner mitochondrial membrane. It is rich in proteins and the ratio of lipid to protein is 0.27 to 1. Hence, it is virtually impermeable to polar and ionic substances. Inner membrane is folded into multiple incomplete septa-like structures called as cristae, which is rich in many enzymes like cytochromes B, C1, C, A and A3, NADPH, NADH dehydrogenase, succinate dehydrogenase, electron transferring flavoproteins, carnitine, palmitol, transferase, etc. Mitochondrial matrix. The region enclosed by the inner membrane is called as matrix. An amorphous material fills the matrix which contains enzymes involved in Krebs citric acid cycle and fatty acid oxidation. Matrix also contains several strands of DNA, ribosomes, and enzymes for synthesis of proteins coded in the mitochondrial genome. So this is the structure showing that of mitochondria, which shows the outer membrane, inner membrane, and intermediate space that is in between inner and outer, and the matrix which is inside the inner membrane, which also contains enzyme complexes, DNA, and ribosomes. Functions of mitochondria. The inner membrane contains a cytochromes of electron transport system and the associated enzymes for oxidative phosphorylation. The TCA cycle takes place in the matrix. The initial breakdown of proteins, carbohydrates, and fats occurs in the cytoplasm of the cell, and the final end product that enters the mitochondria as acetyl-CoA combines with oxaloacetate to form citric acid. The end products of citric acid cycle, that is Krebs cycle, are carbon dioxide, water, and ATP. Like most components of the cell, mitochondria have a short lifespan and are constantly renewed, as they have strands of DNA and they are capable of self-replication as well as protein synthesis. These are some of the mitochondrial enzymes. Membrane enzymes that of outer membrane are cytochrome B5 and B5 reductase, fatty acid co-synthase, phospholipase A, nucleoside diphosphorkinase. Enzymes that are inner membrane are cytochrome B, C1, C, A and A3, NADPH dehydrogenase, succinate dehydrogenase, electron transferring flavoproteins, beta hydroxybutyrate dehydrogenase, carnitine, palmitol transferase, and all translocases. Enzymes that of inner membrane space are adenylate kinase, nucleoside diphosphorkinase, sulfite oxidase, and enzymes of the matrix are pyruvate dehydrogenase complex, citrate synthase, isocitrate dehydrogenase, malate dehydrogenase, fatty acid oxidation system, ornithine transcarbamylase, and alpha oxoglutarate dehydrogenase and acotinase. Golgi apparatus. The Golgi apparatus is also known as Golgi complex or dicytosome. It is present in all cells and generally is located close to the nucleus. It appears as a flat disc that are dilated peripherally and stacked together in a bunch as a stack of cisternae. Anatomically and functionally, it is closely associated with endoplasmic reticulum. Functionally, Golgi apparatus has two main regions, cis and trans. The region nearer to the nucleus is the cis phase, and the region close to the membrane is trans phase, that is trans Golgi. So this is the structure of Golgi apparatus, which is a folded structure which are called as cisternae, and the structure facing towards the nucleus is called as a cis phase, and the structure which is facing the membrane is called as trans phase. Major functions of Golgi apparatus. It is the site for packaging of secretory products into the secretory granules. Materials produced in the rough endoplasmic reticulum travel through the lumen of smooth endoplasmic reticulum. Vesicles budding off from the smooth endoplasmic reticulum transport materials to cis phase of Golgi complex. Some proteins are phosphorylated here and then pass to the trans phase, where they are packaged into the secretory vesicles. It is the site for the incorporation of carbohydrates into newly synthesized proteins to form glycoproteins. And lysosomal enzymes are formed here in the Golgi apparatus. It also transports material to the other organelles and cell surface. Endoplasmic reticulum. Endoplasmic reticulum consists of a network of anastomosing membranes, membranous tubules, vesicles, and flattened cisternae. The membranes of endoplasmic reticulum are continuous with the outer membrane of nucleus and are also connected with Golgi apparatus. Endoplasmic reticulum is the site of the synthesis of proteins and lipids for the membrane of the cells and organelles, and secretory vesicles of the cytoplasm. There are two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum. When surface of endoplasmic reticulum is studded with the ribosome, the organelle is called as a rough endoplasmic reticulum or granular endoplasmic reticulum, as it gives a rough or granular appearance to it. In some cells, such as red blood cells, the ribosomes lie free in the cytoplasm. Rough endoplasmic reticulum are present in more number in cells that are actively involved in protein synthesis, like acinar cells of pancreas and neurons. In neurons, the Nissel's granules are modified rough endoplasmic reticulum. So this is a structure showing the endoplasmic reticulum, which is very close to the nucleus. The endoplasmic reticulum studded with a ribosome, they are called as the rough endoplasmic reticulum, and the endoplasmic reticulum devoid of ribosomes, they are called as smooth endoplasmic reticulum. Functions of rough endoplasmic reticulum. It is concerned with protein synthesis. Therefore, it is abundant in cells of endocrine glands and cells secreting digestive enzymes. It also plays important role in the conjugation of carbohydrates with proteins to form glycoproteins, a function which it shares with the Golgi apparatus. Smooth endoplasmic reticulum. When ribosomes are not attached to the surface of endoplasmic reticulum, the organelle is called as smooth endoplasmic reticulum or agranular endoplasmic reticulum, as its surface has a smooth or agranular appearance. Functions of smooth endoplasmic reticulum. It is concerned with the synthesis of lipids. Hence, it is abundant in cells that synthesize cholesterol, steroid hormones, and phospholipids. In muscles, it is called as sarcoplasmic reticulum, which is specialized for the storage of calcium ions that is released during excitation contraction coupling. Smooth endoplasmic reticulum is part of intracellular transport system, as it is continuous with the rough endoplasmic reticulum and Golgi apparatus. It is also the site for the detoxification or neutralization of hormones and toxic substances. Ribosomes. Though ribosomes are usually present on the surface of endoplasmic reticulum, they are also present as a free organelle in the cytoplasm. They contain about 85% of the RNA of the cell. They are the major site of protein synthesis. RNA is also present in the nucleus, mitochondria, and cytoplasm, and are consequently protein synthesis occurs in these structures too. They may be present singly, which are called as monoribosomes, or in groups or polyribosomes. Peroxisomes. Peroxisomes are the small spherical organelles having diameter of around 0.5 microns and therefore denoted as microbodies. The specialities of uh, peroxisomes are they are formed by budding from or by division of smooth endoplasmic reticulum. They are referred to as subcellular respiratory organelles, but they do not have energy coupled electron transport system. They contain oxidases that promote oxidation of lipids, especially beta oxidation of long chain fatty acids, forming acetyl CoA and hydrogen peroxide, and catalases that liberate oxygen from hydrogen peroxide. They protect the tissue from oxidative stress. With the help of peroxins, the protein chaperones, various proteins with a specific signal sequence, are directed to peroxisome. The membrane peroxisome contains a number of specific proteins that transport substances between peroxisome matrix and cytosol. Peroxisome matrix contains more than 50 enzymes that are involved in many metabolic reactions of lipids, that is, long chain fatty acids, plasminogen, cholesterol, and bile acids, and purines, amino acids, and hydrogen peroxide. Deficiency of enzymes lead to many diseases. Proliferation of peroxisome is caused by many synthetic products that act on the receptor in the nuclei cells known as peroxisome proliferation activated receptors, PPARs. PPARs are members of nuclear receptor superfamily, those on activation to bind DNA and produce changes in mRNA production. PPARs have extensive physiological effect and affect many tissues and organs. So this is the structure of peroxisome with a plasma membrane and crystalline core inside. Lysosomes. Lysosomes are membrane-bound spherical organelles that contain variety of hydrolytic enzymes meant for intracytoplasmic digestion. More than 40 different lysosomal enzymes, which are called as lysozymes, have been isolated. Some important lysosomes are listed. Uh, Lysosomes are found in almost all animal cells except erythrocytes. They are particularly abundant in cells having high phagocytic activity, such as neutrophils and monocytes. In granulocytes, the lysosomes appear as cytoplasmic granules. Lysosomal activity of a cell tissue can be determined. Some of the important lysosomal enzymes, they can be of proteolytic, they can be lipolytic, they can be carbohydrate splitting enzymes, nucleic acid hydrolyzing enzymes, and other enzymes like acid phosphatase and catalases. Some of the proteolytic enzymes could be cathepsins, collagenases, and elastases, whereas lipolytic enzymes are lipases, phospholipases, fatty acyl esterases, whereas carbohydrate splitting enzymes are alpha glycosidase, beta galactosidase, hyaluronidase, and aryl sulfatase, whereas nucleic acid hydrolyzing enzymes are ribonuclease and deoxyribonucleases. So this is the structure showing the membrane and the hydrolytic membrane and the transport proteins of lysosomes. There are different types of lysosomes. There are three forms of lysosomes, that is primary lysosomes, secondary lysosomes, which are also called as endolysosomes, and tertiary lysosomes, which are called as phagolysosomes. Primary lysosomes, they are formed with hydrolytic enzymes synthesized by rough endoplasmic reticulum and packaged in the Golgi apparatus. They are generally referred as storage vacuoles. Whereas secondary lysosomes, these are lysosomes fused with endosomes. Hence, they are called as endolysosomes. Tertiary lysosomes, these are the autophagic vacuoles or autophagosomes. They are formed by fusion of phagocytic vacuoles with the primary lysosomes. Hence, they are called as phagolysosomes. This is in brief about cell organelles. Thank you.